Are you finding it difficult to decide which genes to include in your germline or hereditary panel? You're not alone. It's a complex task, and there are many variables to consider, including assessing the clinical relevance of genes and mutations associated with your phenotype of interest, staying current with the constant changes in hereditary genomics and frequent reclassifications so clinicians can adapt your panels to reflect the latest findings, and organizing the ever-increasing pool of clinically relevant public literature and evidence to determine the best variants associated with your phenotype or region of interest. Hi, Caitlin here again, and welcome to part 2 of our Database Informational Video Series. In this video, we'll explore how HGMD addresses the complexities and hurdles scientists and clinicians encounter in creating accurate and comprehensive panels for germline mutations, and demonstrate how HGMD can save you time when doing just that. But first, a little bit about HGMD Professional, or HGMD Pro. HGMD Pro systematically gathers all known published genes responsible for causing human inherited diseases and disease-associated or functional polymorphisms. HGMD Pro is continually curated and updated to include the latest variant information and relevant literature. It also includes variant classifications, which indicate the pathogenicity or clinical significance of each mutation. These classifications help guide clinical decision-making. Overall, HGMD Pro is a known, trusted resource that provides valuable clinical genomics information to researchers, clinicians, and geneticists. So let's dig in. In this next portion of the video, we'll demonstrate exactly how you can use HGMD Pro to design the best possible cutting-edge germline panels. All right, so let's get started. One way to use HGMD Professional for panel design is to look at the genes that are associated with your phenotype of interest. Here, we can type our phenotype of interest. Let's use craniosynostosis, and then hit Submit. Here, you can see all the different genes associated with your phenotype of interest. So if you're designing a panel, you could include all of these genes. If you're optimizing a panel, you can utilize this information to make sure that the genes that are most up to date in regards to your phenotype are included in your panel. Or you can click on a gene like FGFR2, then click on your phenotype, craniosynostosis, to verify that your panel has the most up-to-date mutations associated with your phenotype for this specific gene. So here, we can see all the relevant FGFR2 mutations associated with craniosynostosis. And you can also look at the key literature associated with those mutations. Now, some users will also use the advanced search option for panel design. And so here we're going to use the Nucleotide Substitutions Plus option. These parameters provide you with the flexibility to identify different and more specific types of variants that you're interested in. For example, say you're designing a panel, and you don't just want to focus on the exonic region, but you may also want to go towards the intronic region. You can define that here. And likewise, you can use any of these other advanced search features to get more specific and granular in the exact types of variants that you want to identify. If you want to look at all the variants that are available, you can simply scroll down to the Other section of this interface and type in your phenotype of interest and then click on Submit. The output provides you with a list of different variants, and so if you're doing targeted sequencing by looking at the coordinates, you can use this to get an idea of what target regions to focus on. Or, if you're working with a microarray, you can use this information to make sure that you have the appropriate SNPs that you want to detect in your specific assay. Now, you can also use this output for panel optimization. So here you can do a cross-reference of what is in your panel with what is listed here to verify that your panel is detecting the most obstinate variants that are relevant for your phenotype of interest. This feature allows you to either create a panel or verify that your panel is up to date with those variants that are known to be associated with your phenotype of interest. Again, highlighting that HGMD Professional is a great tool for panel design by either searching for your phenotype of interest or by looking at those genes or searching for those variants that are associated with your phenotype of interest. Thank you so much for joining us today. We hope this discussion has shed light on how the wealth of genetic information, real-time updates, and variant classifications in HGMD Pro simplifies the complex process of panel design for germline mutations. If you're interested in learning more, you can schedule a consultation call with our experts by replying directly to the email that led you here. And stay tuned for the next part, where we'll discuss how you can seamlessly integrate HGMD Pro into your existing pipelines and what this means for your clinical reporting.
QIAGEN.
Sample to insight.